My name is Nathawani Chuhoithong. My name is Varachaya Phanimek. We are from Princess Julapon Science High School, Drang. Today, we would like to present study of Fiddler crab density affecting soil properties and carbon sequestration in Ban Motamoy Mangrove Forest, Kantan District, Drang Province. Climate change is mainly driven by the continuous increase in atmospheric carbon dioxide, which has caused serious impacts on ecosystems worldwide. As shown in this graph, carbon dioxide concentration in the atmosphere has steadily increased over time, indicating the growing severity of global warming. Mangrove forests are considered one of the most effective natural carbon sinks because they can store large amounts of carbon, particularly in their soils. This ability is influenced by interactions between physical, chemical, and biological factors in soil environment. One important biological factor is fiddler crabs. Their burrowing activities can alter soil structure, improve aeration, and influence processes related to carbon accumulation in deeper soil layers. However, the relationship between fiddler crab density, soil properties, and carbon sequestration is still not clearly understood. Therefore, this study investigated how fiddler crab density affects soil properties and carbon sequestration in Ban Motamoy Mangrove Forest. Question: First, is the density of fiddler crabs related to soil properties in mangrove forests? If so, how? Second, does the density of fiddler crabs affect the amount of carbon sequestration in mangrove soil? If so, how? Hypothesis. First, the density of fiddler crab populations affects soil properties in mangrove forests. Second, the density of fiddler crab population affects the amount of carbon sequestration in mangrove soil. Objective: First, to study the effects of fiddler crab density on soil properties in mangrove forest. Second, to compare the amount of carbon sequestration in mangrove soils in areas with different density of fiddler crabs. Material and equipment we use in this study as follows. Methodology: First, study area selection. Second, division of study areas. Third, soil quality analysis. Fourth, data recording. Fifth, data analysis. Section 1. Study area selection. We select Ban Motamoy Mangrove Forest, Kantan District, Trang Province. The study area was divided in two areas with 5 replicate samples collected from each area. Each subsite was designed as a 10 by 10 square meter study area. Second 2.1 study sites. The first site, Site A, is near the community enterprise and Site B is near the pier. Section 2.2 Field data collection. First, dividing the study area into 2 sites as we stated before. Second, random sampling of 5 quadrants into 2 sites. Third, collecting samples. We separate it into 2 parts. Part 1, recording number of fiddler crab burrows, and Part 2, collecting soil samples. Section 3, soil quality analysis using the group soil protocol. We analyze and collect the data of pH, temperature, soil structures, moisture content, soil color, NPK, percentage of loss of ignition, and percentage of organic carbon. For percentage of humidity, we bake the soil at a temperature of 95 degrees Celsius for 24 hours, then when the dry wet. And then we use the equation as follows. For analyzing the amount of carbon storage in the soil, we take 50 grams of baked soil burned at a temperature of 450 degrees Celsius for 4 hours, left to cool and wet after burning. Then we calculate the percentage of alkali. Then we compare the values to choose the percentage of OC formula. Section 4. Group data entry or database recording. These are the data we have recorded in the database. Fifth, Section 5. Experimental results. Table 1 shows the density of Fiddler crabs and soil properties at each study site. The results show that Site B has a higher Fiddler crab density than Site A. Regarding soil properties, soil moisture was higher at Site B, while soil temperature was higher at Site A. The two sites have different soil textures, but similar soil pH, and soil nutrients were found at only trace level in both areas. In terms of soil organic matter and carbon content, Site B showed higher values than Site A, indicating a greater carbon sequestration potential in areas with higher Fiddler crab densities. Section 6. Conclusion and discussion. In summary, this study shows that Fiddler crab density is closely linked to soil conditions and carbon sequestration in mangrove forests. Areas with higher crab density have better soil physical properties, particularly higher soil moisture and improved soil structure, which are influenced by crab burrowing activities. These activities increase soil porosity and promote water and air movement with the sediment. In contrast, soil chemical properties such as pH and nutrients show little difference between sites, indicating that Fiddler crabs mainly affect soil structure rather than the short-term chemical changes. Most importantly, areas with more Fiddler crabs had higher soil organic matters and carbon content, suggesting that greater carbon sequestration potential. Therefore, Fiddler crabs act as ecosystem engineers, enhancing soil conditions that support long-term carbon storage and strengthen the role of mangrove forests as natural carbon sink. Practical benefit. First, to understand the role of Fiddler crab density in influencing soil properties in mangrove forests. Second, to understand the role of Fiddler crab density in affecting carbon sequestration in mangrove soils. Third, to provide guidance for mangrove resource conservation. And fourth, to support approaches for creating natural carbon sequestration sources. Acknowledgement. We would like to thank the Institute for the Promotion of Teaching and Science and Technology, Mottanoi Community-Based Tourism, directors and teachers of Princess Jalapan High School Drama. These are our citations. And with that, we leave you with a quote inspired by Fiddler crabs. A crab that digs a hole so deep, carbon in the soil it helps to keep. Thank you for your kind attention.